what is the way we classify viruses? And essentially it's, if you are trying to classify them the same way that you classify living organisms, it doesn't work because there is no common ancestor. So therefore there's no tree that you can make. So instead, what David Baltimore said was, why not classify them based on their genome type? Because at least that way you can characterize some of the basic steps in their biology. Because we know that for an organism to kind of function in the cell, the functional part of the cell is going to be a protein. And to make proteins, you need messenger RNA. But based on what you start with, the way you end up getting to messenger RNA is going to be slightly different. And so if we list out the various steps in the viral infection cycle, right? So we have, let's say, attachment, penetration, uncoating, and then early genes. And here is where essentially the Baltimore system comes into play. Because if you are making proteins, so you're expressing genes means that you're taking, in a cell, you're taking DNA, turning it into messenger RNA, and turning that into proteins. But in a virus, you don't always start with DNA, and therefore these steps are not always the same. Now the last two are going to be the same. You're always going to get to messenger RNA, which is going to be read by the ribosome and turned into a protein. And that part has to be the same because the ribosome is the only thing that makes proteins. But depending on how you start, the way you get here is going to be slightly different. And that's all the Baltimore system is based on. So if you're a type 1 virus, your DNA, so this step is easy because it's how a cell works. If you're a type 2 virus, your DNA, so it's really easy because that's how a cell works. So those two viruses work the same way as cells. But if you're an RNA virus, now you need things that cells don't do. You have to take RNA and transcribe that into more RNA. And cells don't do that. And so therefore, the way an RNA virus gets here is going to be based on having an enzyme that no cell has. And therefore, it has to make that enzyme on its own. And so there are essentially these little rules that we can derive out of the Baltimore system simply because it's based on their original genome type. And for our purposes, that's more than good enough. I'm not going to have you draw this where you start with whatever and get to that. It's more important that you understand the general concept of why it works and therefore what are some of the rules that come out of it.
Okay.
And so that's where the rules go, right there. I mean, right.
Yeah, they affect this step
Of
getting from the genome to the RNA steps.
Yeah. I thought we had them in mind. So is there any pictures or anything?
There are probably two questions that you could easily answer with a picture or you could answer in words.
Immunology, MHC class 1, or the Goldfin, can you draw that?
Which one?
The ingest and digest, with the macrophage and the MHC class 1.
So making the MHC complex or actually?
Yeah.
Okay.
I gotta know how to draw that picture properly.
So the MHC complex is a display case on the surface. If we're looking at type 1, this is made by all cell types, and it's based on the idea that the job of a cell is to function, which requires proteins. So we have DNA in the cell, that DNA is going to turn into RNA. That RNA moves out to the cytoplasm, where a ribosome finds it and turns it into a protein. So our cell is full of functional proteins. But over time, those proteins break down, or they wear out, and so we have an organelle called a lysosome. And the job of this organelle is to essentially recycle parts of the cell that are broken down. And it does that by breaking down proteins into the amino acid building blocks. As it does so, it is also going to present some of these protein fragments on the surface in the form of the MHC. And the purpose of doing that is that suppose this cell gets infected, where the virus now penetrates and uncoats, and it makes early genes, or it makes viral genes. So now it's going to make viral proteins. Over time, those viral proteins get broken down, and those viral proteins get displayed on the surface. And so, if the immune system comes along and it's scanning this cell, it'll see that viral protein instead of the normal human protein, and therefore it knows that this cell must be infected. There's also a second type called a type 2 MHC, which is specific for antigen-presenting cells or phagocytic cells, like our macrophage. So what that does is macrophages are phagocytic, meaning that they can ingest things, including bacteria. So let's say this macrophage recognizes a camp from this bacteria, like the cell wall. Well, now it knows that the body is being infected, and so its job is to ingest and digest. But after it ingests and digests, it is also going to display that on MHC class 2. And so class 2 is going to be recognized by T helper cells. And when a T helper cell recognizes a foreign class 2, it is going to activate a T killer cell. And a T killer cell comes along, finds infected cells, and kills them off. Now, in this diagram, I have a blue and a red different antigens, so not quite the same, but the general idea is the same. Other questions or concepts we'd like to go over in more detail?
Was there anything else?
Just the
Ask him. Now's the perfect time.
This test should be much easier than the first one.
Do we
Kind of
even
always
know
saying,
how
just
to
have
Oh,
to
yeah,
find
we-
the first.
Yeah, you can draw a virus? Yeah, we're gonna have to, that's gonna be easy.
That's easy.
There it is. So assuming it's probably gonna be that.
You keep saying this one's gonna be easier. How was the very last one, in your mind? How's that?
So it's weighted more.